Welcome to DoveBiology, Bio Lessons to Go. In this video, we'll be exploring meiosis and the origin of genetic variation for AP Biology. What is genetics? Well, genetics is the branch of biology that studies heredity. Genetics will look at traits or characteristics that are passed from parents to offspring. You may have noticed that offspring oftentimes resemble their parents. Well, this is because they inherit genes from their parents. These genes act as a set of instructions that will determine the traits of that organism. The genes themselves are made up of a sequence of DNA nucleotides that will be part of a chromosome. Each species will have a unique number of chromosomes and therefore have a unique amount of DNA. For example, a typical human body will have 46 chromosomes or 23 chromosome pairs. Each chromosome in the pair will have a twin. You'll inherit one of those chromosomes from your mother and the other from your father to get 46 total chromosomes. The two matching chromosomes are referred to as homologous chromosomes, where homo is a prefix which means same and logos is a suffix referring to shape, because homologous chromosomes will have the same size, shape, and appearance under the microscope. These maternal and paternal chromosomes in a homologous pair will have the same genes at the same locations, but possibly different versions of those genes. You might have a gene for widow's peak on the maternal chromosome, but a gene for no widow's peak on the paternal chromosome. And because widow's peak happens to be dominant, that's what shows up in your phenotype. Now, in order for two parents to give equal amounts of genetic material and then produce a child with the same amount of chromosomes as themselves, each parent must give half of their genes. If instead they gave all of their genes, the chromosome number would actually double with each subsequent generation, providing offspring with way too many chromosomes than are necessary for the traits that they need to inherit. In order to pass on half of their genes to their offspring, the parents will produce specialized cells to do this, called gametes or sex cells. Males produce sperm, while females produce eggs. These gametes will have exactly one half of the normal chromosome number. Because they have half the chromosome number, we say that these cells are haploid. In humans, the haploid number will be 23. In the process of sexual reproduction, the nuclei of these gametes will join together in a process called fertilization. The resulting cell, a fertilized egg, is called a zygote. After fertilization, the zygote and all of the body cells that come from the mitotic division of that zygote will have two sets of chromosomes. Because the zygote and the cells that follow have two sets of chromosomes, we say that they're diploid. So the diploid number for the typical human is going to be 46 chromosomes. So to recap, gametes are simply sperm and egg, and they are haploid cells. They have half the chromosome number. The zygote, on the other hand, is our fertilized egg. The zygote results from the fusion of one sperm and one egg to produce a diploid cell that contains two sets of chromosomes. Now, to produce these haploid cells, we need a specialized process, and that specialized process is called meiosis. Now, meiosis is very similar in a way to a previous process that we talked about, mitosis, in that they're both forms of cell division. However, there are two distinct differences. Mitosis is a single division that results in two identical body cells during the process of growth and repair, whereas meiosis is two separate divisions that will result in four haploid sex cells that are prepared for sexual reproduction. Now, the alternation between meiosis and fertilization is common to all organisms that reproduce sexually. But there are actually three different types of sexual life cycles that we'll see that differ in the timing of meiosis and fertilization. For example, if we look at animals, the typical animal is diploid from the time that it's formed with a zygote all the way throughout its adult life. The only cells in an animal that happen to be haploid are sperm and egg that are produced by meiosis. Plants and some algae, on the other hand, have what's known as an alternation of generations, where at some point in its life cycle there are diploid cells, and at other points in its life cycle there are haploid cells. Starting from the zygote, which would be diploid, the zygote would develop by mitosis into a diploid stage of life, which we call the sporophyte. The sporophyte is responsible for producing the haploid gametophytes through the process of meiosis. The gametophytes, haploid, will divide by mitosis and maintain their haploid state until the point at which they produce the gametes, which then can fuse to create our diploid zygote and continue that sexual life cycle again. Fungi and protists also have an alternation of generations. The difference is the bulk of the life cycle of a fungus is actually haploid. When you're looking at a mushroom, for example, you're looking at haploid cells. The haploid body cells of a fungus will divide by mitosis. At some point in its life, two of those haploid cells may fuse to form a diploid zygote. And that's the only time in the life cycle of a fungus and some protists that will have a diploid cell. That diploid zygote will actually divide by meiosis to create the body cells for that fungus, which will then continue to divide by mitosis until it's time to reproduce again. So really, depending upon the type of life cycle, either haploid or diploid cells can divide by mitosis to create identical cells. But only diploid cells can undergo meiosis. In all three life cycles, though, the halving and doubling of chromosomes will actually contribute to the genetic variation in the offspring. So the meiotic processes is very important in creating that genetic diversity. To do that, we'll have to examine that process more closely. Meiosis consists of two major stages called meiosis 1 and meiosis 2. Meiosis 1 is the first division in this process, and it's often called reductive division because it's during this process that the chromosome number is halved. Meiosis 1 begins with interphase 1, where we look like we have kind of a normal cell with a nucleus that's fully intact, and the genetic material is thread-like chromatin. Now, at some point, that cell will have a signal that it's going to need to divide, and so it will enter into S phase of that interphase. Now, during that S phase, the cell is going to copy its genetic material, and we're going to have DNA synthesis, and the chromosomes will replicate. Now, the replicated chromosomes will consist of two identical sister chromatids. So if we look at this example, we have some red chromosomes and some blue chromosomes. The reds from our mother and the blues from our father. The chromosomes of similar sizes would be homologous chromosomes. But the replicated pairs, the two blues of the same size or the two reds of the same size, are what we call sister chromatids. And these sister chromatids will be held together at the center called a centromere. So our homologs, when they're duplicated, will consist of pairs of sister chromatids. Now, in prophase 1, these replicated sister chromatids will actually match up with their homolog and form a structure called a tetrad. Now, because these homologous chromosomes are close together and they share genes in the same location, they oftentimes will exchange genetic information with each other to increase the diversity in the offspring. And this process is called crossing over. Now, after crossing over, the nucleus will have disappeared and our tetrads will attach to our spindle fibers. In this case, the tetrads will be pushed and pulled to the middle of the cell, to the equatorial plate. The reason the tetrads are aligning at the equator during metaphase 1 is to make sure that we're going to be able to reduce the number of chromosomes by half when these tetrads split. So instead of lining up individual sisters like we see in mitosis, the tetrads are aligning at the equator. So that during anaphase 1, the tetrads are going to split and go to the poles. This allows for one set of chromosomes to go to one pole and the other set of chromosomes to go to the other pole. And that will create two cells, each with one half the original genetic information. We've had reductive division. We divided the chromosome number by half. Now, the reason why we don't stop here is because even though the cells are haploid, they're still doubled up. They still exist as sisters. So the whole process of meiosis 2 is all about splitting the double-stranded sisters. So in meiosis 2, we're not actually reducing the chromosome number, we're just separating the double-stranded sisters. Meiosis 2 in humans will begin with two cells that have 23 double-stranded chromosomes each, and it will end with four cells, each with 23 single-stranded chromosomes. Now, the process of meiosis will allow for us to form our sperm and our egg, but there's a slight difference between the formation of sperm, spermatogenesis, and the formation of egg, oogenesis. In spermatogenesis, a diploid primary spermatocyte will divide by meiosis in meiosis 1 to create two haploid cells, which we call secondary spermatocytes. These secondary spermatocytes will divide by meiosis 2 to produce four haploid sperm cells that will mature and form, have the tail grow, and then they will be able to be used in sexual reproduction. Now, when we're forming eggs in the females, our primary cell is called the primary oocyte. It will divide by meiosis 1 and create two cells, but there will be something different that's happening here. In that first division, there will be an unequal distribution of cytoplasm, such that one cell will receive more of the cytoplasm than the other. The cell that gets the bulk of the cytoplasm is going to become our secondary oocyte. And is going to be our egg that will be used eventually in fertilization. Whereas the other cell that has less of the cytoplasm is going to become what we call a polar body, and that will be discarded by the female. Now, in mammals, the secondary oocyte doesn't undergo meiosis 2 right away. Instead, it waits until after ovulation, and as it passes through the fallopian tube, it waits to be fertilized. Once the secondary oocyte is fertilized in the fallopian tube, it will undergo a second meiotic division, producing another polar body. The new polar body will also divide so that we'll end up with four cells at the end, but only one of those four cells will serve as the egg, and it's a fertilized egg. Now, with the exception of identical twins, children in the same family are never alike, which is kind of strange because they're coming from the same set of parents. Now, the reason why these children, like the Baldwin brothers, have genetic differences has everything to do with the details of meiosis 1. So the first source of potential division occurs during crossing over. During that prophase 1, when the tetrads form, the homologous regions of the tetrads can actually exchange with each other as a result of overlapping at regions which we call chiasma. As a result of this crossing over event, we have genetic recombination that's occurring, so that the offspring may actually have combinations of genetic traits that neither parent actually had. The second opportunity for genetic variation comes during metaphase 1 of meiosis. The orientation of those tetrads at metaphase 1 happens to be a matter of chance. Each chromosome pair, when they arrive at the equator, will orient itself independently of the other pairs, such that every time meiosis occurs, the lining up can actually be different. And this is known as independent assortment. The chromosome pairs, the tetrads, are aligning independently of one another. In humans, as a result of this, there are more than 8 million different possible arrangements. With 23 tetrads arriving at the equator, there are 2 to the 23 different possible arrangements. So when we look at this example, we see that we have a cell that has 3 pairs of chromosomes. Each time that they arrive at the equator, we get a different combination. And with only 3 pairs of chromosomes, we actually end up with 8 possible outcomes. So this shows us why meiosis is such an important process. Meiosis allows us to maintain the normal species chromosome number by preparing haploid gametes. Since each parent gives half their genetic information, it ensures that the subsequent generations have exactly the same number of chromosomes as the previous generation. And then, of course, as a result of things like crossing over and independent assortment, meiosis has the opportunity to increase that genetic variation by recombining genes in the eggs and the sperm that we didn't see in previous generations.